Back to the session. The next speaker is Jenny Larsson from the University of Sheffield, and she talks about the shell shape of snails.
Yes, thank you. And before I start, I would also like to thank all the organizers for arranging this workshop. It's been really nice being here, and also all the previous speakers. It's been really interesting hearing what everyone has been up to. Yes, I will also like to thank my two supervisors, Roger Butlin, who's an evolutionary biologist, and Torbjörn Lund, who's a mathematician. So I'm going to talk to you about my PhD project. It's on using landmarks from images to obtain growth parameters for snail shells. So this is going to be another talk on biological growth and form, but from a mathematical perspective. And one of the good things with working with snail is that you can have all these nice images. So I hope that you will enjoy this presentation.
Sure.
Yes, so first I'm going to introduce you to my species. This is the small marine snail, Littorina saxatilis, known as the rough periwinkle. It's very common all over the North Atlantic. It lives on rocky shores, and the ones that we have here are from the Swedish west coast on the top and the Spanish Atlantic coast on the bottom. And one of the interesting things with this species is that it consists of two ecotypes. That means that we have two types that are adapted to different habitats. And in this case, we have the crab ecotype, this one, who's evolved a bigger, thicker shells with smaller relative aperture to in response to being having crab predation. And we have the wave ecotype, which has evolved a smaller, thinner shell with bigger relative aperture as a response to wave action. There are often very sharp transitions between these two habitats, meaning that the two different ecotypes can exist in distinct habitats just a few meters apart. And in this contact zones we also have intermediates, so they are still the same species, but they they're separated into these two ecotypes. And this separation has been studied a lot in biology, mostly in the context of speciation and parallel evolution. And because of this, their genome is being sequenced, so it might be possible to get to know which genes are related to different types of growth. And yeah, as some of you have already mentioned, one of the big things in biology now is to connect the phenotype with the genotype to understand what the genes do with morphology. But up until now, the methods used to describe the shape difference has mostly been involving the Procrustes method and landmarked to the images. And the result there do describe the shape differences, but it doesn't really relate that well to the construction process of the shells. And the shells as whole 3D structures, which is the reason why we want to build on Raup's growth parameter models to understand this better. Yes. So some of you might already be familiar with this, but I'm going to give a short explanation of the Raup's growth parameter model anyway. So we basically have a logarithmic spiral that rotates around a vertical axis, and then we have around this we have a generating curve which sweeps out this surface. In this case it's just a circle as just a simple version. And this model consists of 3 different exponential functions. And they are related to the height of the spiral, the radius of the spiral, and the aperture size. So first we have, and each of these have 2 parameters. One for the start values, or in this case, end values, which are the values of this spiral, the height and radius of it at this endpoint, and the aperture size at this point. The other parameters are the growth parameters. And they are the difference in height, radius of the spiral, and aperture size from one turn to the next. Yes, and in this case, I've just plotted a simple one, but all of the growth parameters are about 0.09, which means that they will be, because of the exponential function, relate to an increase in size by 76%. So from each turn to the next, we will have an increase in height, radius, and aperture size by 76%. But yeah, this is a fairly simple way of modeling shape. And it relates very well to the construction process. So it's thought that this would be a good way of understanding the shape differences between different types. Oh yeah, and in my visualizations I will also add an elliptic lower part to the aperture. So the upper half is still a circle, because this is a simple way of extending the possible aperture shape with adding just 2 parameters, one for the eccentricity of the lower part and one for how it's rotated. And we also have measured values for the thickness, so that I also have in the visualizations. But those those parameters are not part of any statistical analysis yet. So, in addition to using the shells to verify that we actually have the correct growth parameters, we can also use them to understand the internal structure without having to scan them, and that saves a lot of time and money. And more importantly, since the original shells are destroyed during the biological analysis process, we don't have them anymore, and this would be a simple way of reconstruction. And yeah, they could also be used for fluid dynamics. We could see how changes in shape will affect how well they fit in the wave-swept habitat, or we could do shell strength analysis to see how well they resist crab predation, and that would mean that we get even more information about these growth parameters and relating them to the different selective pressures. So this is what I have. I have photos of the different shells, and we would like to reconstruct 3D shapes from 2-dimensional data. And I have a lot of these images. They've been collected for a long time, and they've been using the same way of positioning them and photographing them and landmarking them. So I have landmarks for all of them, like this. But the position of the shells and the way the landmarks are placed are done with the Procrustes method in mind, so they might not be ideal for doing growth parameter extraction. So there are some problems with this, but I will still still try to infer the growth parameters from the data. And one thing is that these landmarks are not. We have one that is truly homologous. It's the apex, so the top point on all of them. They are homologous, but the other ones are not really, because even in the same shell over time, when it grows, you add more material along, so it gets a new aperture, and then you would have to rotate it, so they won't be exactly the same point. But yeah, it's possible to do something with it anyway. I do so making use of this data, I do some assumptions that are not necessarily true. For example, I say that these suture landmarks are going to be on the widest part of the on this whirl, and this will be on the widest part of this. And yeah, it's not necessarily true, but it gives a good approximation of what's actually going on. But using all these assumptions, we can actually get some approximations for the growth values. So I do some geometry and some fitting of a circle to the upper part of the aperture. And the green points are what I fit exponential functions to get the growth parameters. And I also have to align all the images in a good way. And that is causing some problems because we'll talk more about that later. The lower part of the aperture doesn't really affect the growth parameter, so we just add that as an extra thing on top. So it doesn't really affect any of the parameters that we're investigating. And this is the result. Not always, but for a fair amount of them. And I'll talk about some problems later on. And the difference here is, of course, in the size. This is the start values in millimeters. But we're not that interested in that for the moment. We're more interested in the growth parameters, which are also different. Good. Yes, we can see that for the crab type it has lower values for the growth. Might seem counterintuitive because it seems taller, for example, so maybe it should have higher growth values for the height. But it's actually the relative growth that is important here. And if we look at it from the other end, so we start at this end point and then go backwards, it means that we have a faster decrease in height, radius, and aperture shape. This one has the faster decrease because it has larger values, and this one has a slower decrease when we start here and go back. Yes, let's also have a look at them from other angles. This is the nice thing with having the growth models. You can have them in 3D. And these are, of course, some of the better ones. I don't get this good result for all of them, but a lot of them are actually pretty good. And if we have a look at the actual values here, this is somewhat the same as the example that I showed earlier, so it increases with about 80% for every turn, while this one has an increase of about 140% for each turn. So it's quite a big difference in how much they grow. Yes. And now if we look at how shape variation is visualized when we with the previous method using thin plate splines and principal component analysis, the first principal component has actually been used as the way of separating the ecotype into the different types. So this would be at the wave end of PC1 and crab end of PC2. And yeah, these ones are just the ones that I showed earlier. So intuitively, it seems like a good idea to have these to understand how they are different. But yeah, I've only shown you these two examples, and they are pretty good. But how well is this method when we look at a bigger set of shells and do some statistics? So the data I've been using is from the Swedish west coast, and it was sampled before I even started this project. But it consists of boulder field here in the middle, which means that it's a sheltered crab habitat, and on either side we have smooth cliffs, so they are more swept by the waves. So a crab habitat in the middle and one wave habitat on either side. And approximately 600 snails were collected here, and their position along the shore was recorded. So each snail was photographed and landmarked, and there was also some other biological analysis on all of them. Some traits for the animal that actually lives inside, for example. And of course, some genetics. And this is the result so far. On the vertical axis, we have how far along the shore they were collected. And the blue part is the crab habitat, and the green parts are then the wave habitats. We have some down here. It's just they sampled far into the wave habitat to just make sure that they actually got proper ones of that type. Yes, and here I've combined the radius and aperture growth into one total width growth. So how the total width here varies from one to the next, to make it easier to visualize everything. And there seems to be some difference between the habitats, at least. And we can see this even more clear if we project everything down to the height growth with growth axis. So this is what it looks like from above. So the green wave type has bigger values for the growth, and the crab type has smaller values, which is consistent with the two examples that I've been showing. And bigger values means a bigger relative change in the size. So that is what is expected. But how accurate is it? I mean, we might see a linear trend here if we disregard a bit of the endpoints, but there is more variability here. So what's really going on here? So if we look at this again, but we don't have the color from the different habitats, we change it to a goodness color. So how well do the models fit with the actual shell? And the green ones are, of course, the good ones, while the red ones are the ones that are bad. And here it does seem to be some type of linear relation between the types, between the parameters, as we go from one habitat to the other. So there might be something going on there. Need some investigation.
Can I have a question?
Yeah.
So how you check that the accuracy of your 3D reconstruction?
At the moment, it's just me looking at at the two and saying it's good, it's bad, or it's somewhat good. So I haven't done any proper statistics or on it yet, but that is one of the things I want to do, but I haven't had the time, unfortunately. And yeah, these two that we've been looking at are these 2 points. So these two are some good ones, but now I'm also going to show some of the less good ones. Sometimes we get something like this, and that seems to be when these ones will actually be red dots within this green cluster, because the total width gives good values, but when we try to split it into radius and aperture, then we get something wrong. I will say something more about that in a minute. Or we could get something like that, and those are from this outer part here. Because we have some problems with properly aligning the vertical axis. And if we can't align that, then everything else will also not be as good as we want it to. And yeah, the reason for it being less accurate here among the wave type is that the assumptions that we do are more true for the crab type. It's easier to use that one because it has more of a spire, so the suture points are going to be more easily used, while on this one they might be harder to find and less accurate to place for the people doing the landmarking, for example. But if you think of the green ones that give us a good model, I think that this is what it will look like, which parameters we will have when I get the method to work better. Start values. Haven't talked about them in a while. This is the result for the start values. So aperture size, start height, and start radius. And if we also here look at this goodness score, we see that most of the red ones are in this area, which is mainly because of the start radius gets wrong because of this relating to the vertical axis. So it becomes too small, too close to the axis, and then everything else becomes less accurate. So this is something I need to fix. But if we would remove these ones that are not good models, then then there is actually at least no visible difference if we just look at the graphs between the two different ecotypes in the start radius and start height. It's only in the aperture size that we can see a difference relative to the total size then. And yes, this part of the graph corresponds to the bad ones on the other one. So those are the ones that give bad results. So this is definitely something that needs to be fixed. And here are the bad ones, or some of them. And in the first two, you might think that they don't look so bad. Why would they get bad results? But that are these ones. And what I think is happening here is that when we try to align this vertical axis, the growth pattern has actually changed. So from the first few spirals, it grows along one axis, and then something happens and it tilts slightly, and that makes these this split into radial and aperture growth. It doesn't do it in the way I want it to. And of course we have that one which has lost part of its aperture, so the landmark would be here and it wouldn't really represent the true aperture very good. and this one. When it's flat like this, it's really hard to find where is the true apex. Is it here? Is it, yeah? Where on this is the true aperture, or has it completely gone? Because a lot of the time it's get worn away during its life, so the true aperture might even be a bit further away. And all these things relate to this aligning of the vertical axis. But it's something that I'm trying to look into to see if I can fix. So, conclusion. Even with all these problems that I've told you, we can still get an analytic method that gives us the growth parameters. At least on a statistical level, we get a lot of them that are good. And these parameters do describe the shape variation in the way we expect. And they should also be closely linked to the genetics behind it, but that has yet to be investigated. And we do get these nice 3D computer models. And here I've put them into the correct size. So this is the small wave type and the big crab type and a couple of intermediates in between. So these are how big size difference we have. It's we do even get even bigger crab and smaller waves, but yeah, these are there's big variability within this species. Next step. Yes, here is actually a CT scanned shell, which might be useful, but we don't have that for all the shells. This is just a few ones that have no other biological data connected to it, unfortunately. But yeah, next step would be to, of course, improve the accuracy of the method so that we get the correct values for more of them. And here I think some shape analytic method might be useful. I'm looking into it, but I haven't gotten anything else. We're also doing an aperture shape analysis, or we're about to start it. And the thing is, this is an example that has been found in the lab when crossing the different ecotypes. They don't find them in the wild, since they probably can't survive. But they do get them in the lab, and the idea is that maybe this happens because of some incompatibility from these growth parameters. We're also, yes, so having this, when it's good enough, we want to connect it with the genetics. And since this is such a simple method to do, should be easily extended to different shells. This one, for example, looks more like the Spanish crab type that I showed you in the beginning with all the ridging. But it should be easy to extend to anything with a logarithmic growth pattern, essentially. Yes, that was everything. Thank you for listening.
A couple of questions.
I have two questions. The first one is, how do biologists decide whether they're looking at two different species or the same species but different ecotype? For example, your two ecotypes are dramatically different in shape, color, and size.
Yes.
So how do they know?
In this case, but yeah, it's a hard question. A lot of biologists are trying to define what it really means to be a separate species or not. But in this case, there is there's gene flow between the species, so they do interbreed, and the hybrids still lives on and breeds into the next
small ones
generation.
and the big ones interbreed?
Yes, they do.
My second question is, can you apply your methods to shells that don't have logarithmic curve as their basic? And also, where does it come? Why do so many species, for example, the D'Arcy Thompson beloved Nautilus is also logarithmic spiral? Why logarithmic spiral?
I guess it's because it's you have the same increase in growth all the time. So at every time step, you grow with the same same factor. So for one turn, for example, you grow to twice the size, and the next turn you grow to twice of that size. So I guess it's just a easy way of getting the shape, because it's related to the size you have at the moment.
No, but is that interesting? Because okay, geometrically it makes a lot of sense, but you can say that biologically it's not the same, because there is this exponential expansion. So it's consumption of material you need. It's not the same. So you have this.
And also, if you don't have the same growth parameter for all of them, it will only work for. If you have the same parameters for all of them, it should work for all the time. It can grow however big it wants without having too big change. But if there is a mismatch in these growth parameters, it could be that it would start growing too big with a too small aperture. So it would be just a tube that goes separate from everything else, and that doesn't really happen.
When big and small interbreed, what do you get? Big or small, or some probability of big and small?
They have been investigating this in the lab, having the two ecotypes and see what the next generation is and the next generation after this. But I think they actually haven't published anything yet about it, but you get intermediates, and sometimes they're more crab-like and sometimes they're more wave-like. So they are in between.
I was just thinking about the relation between the shape and the parameters of the shape and your landmark points. So it seems to me you have a very good model. So in terms of inverse problems, it looks like you have a very good forward model, let's say.
Yeah.
So is that how you relate? Is that the way to relate the two, or to think of it as an inverse problem? Or?
Well, so far I kind of recently got this thing to work at all. So now I'm trying to do all the analysis to find out how I can fix everything better. But at the moment I'm just taking the landmarks, saying that they are in the mathematically good way, which they're usually not, but good enough approximations, and then just put it into an analytic method to get the parameters. So I don't really do any optimization or anything yet.
So maybe that could be the next step.
Yes.
Something like that.
Yeah, I have a lot of ideas of what I want to do now that I have something that is working somewhat.
Please, let's thank Jenny again.